I don't know if you need to turn the volume down on that one in case it'll echo. Yes, 7 minutes and 3 minutes of question answer. Okay. You good, Adam? Looks good on my end. Looks good, Rita. Okay, thank you, everybody. Thank you for bearing through our technical difficulties this morning, but we have persevered and we are here. There are people who can see us live. There are parents who are here in person, so we're very thankful for that. So the schedule I set out said that you all would be starting at 10:10, but as you'll learn, everybody here at UCLA is always plus or minus 5 minutes. Okay, so we'll get back on track eventually. So we are here today for our Nanoscale Microscopy Summer 2024 final presentations. Ooh. So we are here at UCLA, and we're here at the CNSI. So we are the California Nanosystems Institute here at UCLA. We are a department at UCLA, but we're not a degree granting department. We are instead an interdisciplinary hub of technology, research, education, and entrepreneurship. And so part of what we do is our education team. We do things like K through 12 teacher training workshops. We have a nanovation competition, which incorporates nanoscience and entrepreneurship. We do public outreach demos, and our summer is filled with these summer session programs for high school students. We also have these technology centers here at CNSI. You'll see we have 6 of them, and they all have very long, obscure acronyms. That's also a UCLA theme. And the 3 that we focused on for this week with your students are the 3 up top here. So this is our Electron Imaging Center for Nanomachines, the EICN, the Advanced Light Microscopy and Spectroscopy Lab, the ALMS, and the Nano and Pico Characterization Lab, or NPC. And so your students learned about light microscopy, electron microscopy, and scanning probe microscopy throughout this week. And so each one of these subjects, scientists spend years studying and cultivating their craft in them. And your students learned about all of them in 5 days. So we just want to give our students a pat on the back for persevering through all of this information. You guys are awesome. And today we're going to show you all that they've learned throughout this week. So you'll see that on Monday, we had them build their own microscopes in these DIY microscope sections. They heard some guest lectures. And then Tuesday was all about light and fluorescence microscopy. Wednesday was all about electron microscopy. And Thursday was all about scanning probe microscopy. So again, we were 9 to 5 every day here learning, okay? And we wanted to teach them these skills in science, so skills about nanoscience, microscopy, and being a scientist. Students, I hope you reflect on these questions and see how your answers have changed from the beginning of this course now to the end of this course. And we also want to teach them these skills in life, so things like a growth mindset, critical thinking, communication, collaboration, creativity, curiosity, problem solving, and reflection and introspection. So all of this was, again, jam-packed into our 5 days that we have with your students. And so we want to give a thank you to our nanoscale microscopy instructors. Some of them are here today in the audience and will be questioning the students a little bit more. So the directors of our various technology centers that you heard about, Laurent Bentolila over here, Adam Stieg as well, Hong Zhao, who's in the building. I saw him this morning. He'll probably make his way down eventually, and Matthew Mecklenburg. So these are all of our experts that your students learned from this week. So thank you. We also want to thank our guest lecturers, Professor Yuzheng Li, Professor Jeff Miller, and Artem Goncharov. They all took their time to explain to your students some of their specific research topics that they study and how microscopy is applied in their actual research labs today. So thank you to our guest lecturers. And then we need to thank our teaching assistants, our residence assistants, and our technology center staff. So Jordan Teeter, who's up here in the front, is our teaching assistant. He is an expert in all three of the different techniques. He's shaking his head right now. He's an expert in all of the techniques, so he helped every single day this week with your students in the labs and showing them how things work. And then we have our residence assistants, Sarah, Kylie, and Marlo. They lived in the dorms with your students this whole week. So let's give them an extra round of applause for that. And then our various technology center staff, again from the ALMS, EICN, and NPC, demonstrated how these instruments work for your students. So our afternoon activities were filled with them going down into the labs and really seeing how this technology is used and applied. And so all of these technology center staff, we want to thank for the time that they donated. They got free lunch, but we'd also like to applaud them. Okay, and then some of our staff that's helping us out with all sorts of things. We have IT, events, communications, and our work study students who helped us throughout all week, helping us make sure everything's working properly. So big thanks to them as well. Okay, so these final project presentations, students are going to present a novel research project idea using one of the nanoscale microscopy techniques that they learned this week. So they'll give a 7-minute presentation. Kylie and Jordan here are gonna have big timers here for you all to watch. I'm gonna, cut you off at 7 minutes, and then we'll have 3 minutes for Q&A from the audience. So my audience, I also need you to be good participants. There are no stupid questions, okay? And students, if you don't know the answer, totally okay to admit that you don't know the answer. You just thank them for their question, tell them you'll look it up later, and get back to them, okay? Okay, so audience, I need you to participate as well. Adam and Laurent, go easy on them, all right? Okay, so this is our schedule. Again, we're a little bit behind schedule, but we're gonna have our groups go 1 through 10. We'll have a 10-minute intermission as well, so we can all do a little stretching break, okay? So group 1, come on up. Opposite way, group 1. Okay. So again, your clicker and laser pointer and then phones.
Ladies and gentlemen, good morning, and thank you all for being here. I am Robin.
I'm Yubin. I'm Isa.
And we are honored to discuss about the use of fluorescence microscopy with new techniques to track cancer cells for glioblastoma research. This is a transformative development in the field of cancer treatment. And now let's welcome Yubin to start us off with the abstract.
So a summary of our research idea is that we're working on making a new fluorescence plan that consists of 2 techniques. And this is to help with glioblastoma research. So our 2 techniques we're using is fluorescence sodium and fluorescence in situ hybridization. And these 2 techniques will be used to help with glioblastoma research, which is a very incurable brain tumor.
Fluorescence microscopy has revolutionized the way we approach to cancer surgery. This high-resolution imaging capability offers surgeons real-time visual guidance during operations. Furthermore, one of the most significant advantages of fluorescence microscopy is its ability to differentiate between cancerous and normal tissue. With a clearer understanding of the tumor's boundaries, surgeons can remove the malignant tissue more precisely, reducing the risk of damaging critical areas of the brain. Now, let's talk about the BBB, also known as the blood-brain barrier. The The BBB is a selectively permeable membrane that separates the blood from the brain's interstitial fluid, whereas this protective mechanism also halts the delivery of drugs to treat neurological diseases, which include brain cancer. And by solving this issue, it involves the use of fluorescent sodium, which will later be mentioned.
This is the current state of the glioblastoma research. The primary treatment for glioblastoma is the surgery to remove the brain tumor, and that's where the fluorescent sodium comes in. As we all know, in the case of a healthy individual, the blood-brain barrier is highly selective and it does not allow any dyes to pass through it easily. However, in the case when a brain tumor is forming, the BBB is compromised, where the fluorescent sodium could have the chance to pass through the BBB since it is really small and the barrier is broken. The image illustrates how the fluorescent sodium illuminates the tumor in the surgery, and we could quickly identify and remove the tumor. Researchers has also found some several genetic mutations that associated with glioblastoma, which commonly occur in the EGFR, PTEN, and other genes, since they're really common. And this research could provide a base for us about the potential genes that we could use to track the formation of the tumor.
So our main technique that we're utilizing is fluorescence microscopy, and fluorescence microscopy is a microscopy technique that uses fluorescence to study the properties and structures of substances. So with this microscopy technique, we're utilizing two main techniques, which are fluorescein sodium and fluorescence in situ hybridization. Fluorescein sodium is a water-based substance that dissolves in contact with water. So in this case, it would dissolve in the blood in the brain, and this allows the substance to act as ions and pass through the blood-brain barrier. This technique has actively been used on live mice and is currently being researched to use on patients. Fluorescence in situ hybridization is a technique that uses small molecules of DNA to detect specific DNA sequences in the brain cells.
So our solution, instead of applying the fluorescent sodium during the actual tumor surgery, we would like to use it for the research and treatment in the pre-cancer stage, where we detected the cancer cell, but it hadn't formed the tumor. We propose a research plan aims to study the behavior and the characteristic of the glioblastoma cells. This plan would help us identify the mutations, gene amplification, and other alterations that drive the tumor growth. Eventually, it will help us in the development of the target serotypes, leading to the better treatment outcomes for the patients. Let's dive into how our proposed plans work. I split it into two parts. For the human treatment, we inject the fluorescent sodium into the patient's body. In the case of the tumor, this compound would accumulate into the tumor tissues, and it allows us to visualize the tumor in real time using the fluorescence microscopy. For the research experiments, we plan to inject the FS into the mouse model for imaging, and then we use the technique called the fluorescent in situ hybridization, which is the FISH technique to study the cancer cell groups. And we use the fluorescent labeled DNA or RNA tubes specific to genes that we search on, such as FGFR and PENT. These tubes are designed to blend to their complementary DNA sequences within the glioblastoma cells, which incubes the slides with these tubes under controlled conditions, allow them to attach to their target sequences within the glioblastoma cells. After the hybridization, any excess tubes that have not bonded to the target DNA sequences are washed away. So the outcome for the process is more understanding of the cancer cell regarding to its behavior, which will help us identify the potential targets for the serotype. Following, we combine the real-time imaging and genetic analysis techniques to give us a comprehensive understanding of the tumor behavior and facilitate the development of the target and personalized serotypes.
So the reasons that our research idea is better is for three reasons. It's because it's safe, reliable, and efficient. Our research idea is backed up by techniques that have been studied and researched over the past few years, and they've already been tested on live mice, and they're currently being researched to test on patients. So this is why it's very safe and reliable to use these techniques. It's also efficient because we're attacking glioblastoma research in several ways, including the blood barrier crossing and DNA tracking.
Some of the future research directions could be how does long-term use of fluorescent sodium impact the survival rate and neurological outcomes in glioblastoma patients compared to a traditional imaging techniques? And what are the molecular mechanisms by which fluorescent sodium selectively accumulate in cancer cells? And how can this mechanism be utilized to develop more targeted therapeutic interventions? And the turning point in glioblastoma treatment with advanced technologies like the use of fluorescent sodium have set the stage for several directions. Well, first of all, enhanced visualization and imaging technique. Provides surgeon with real-time visualization of the glioblastoma cell during surgeries or even 3D imaging, aiming to enhance tumor identification and restrictions. And secondly, personalized medicine based on genetic makeup of the tumor. Targeted treatment can be designed for patients that could restrict the growth of the tumor more efficiently. And finally, reduce neurological damage. With the ability to accurately identify tumors' margins minimizes the risk of damaging critical brain areas. And in conclusion, fluorescence microscopy represents a leap forward in the battle against brain tumors. This technology not only improves the patient's outcome, but also offers hope to those battling the most aggressive forms of brain tumors. And thank you all for the attention, and I hope you enjoy the rest of the presentations.
Okay, questions from the audience, including other students.
It seems your proposal, you're trying to target them at the precancerous phase, right? So there are two components, the earlier timing and then also better targeting, right? Do you think that both could have roles in patient outcomes? So how do you know who to target for the earlier screening? Right? Because the symptoms haven't manifested yet. They may not show up at the office yet, right? So how do you do the earlier timing?
I mean, since like brain cancer, which is like brain tumor, it's a type of cancer, it has like different phases. So in some early phases, we could probably use like therapeutic methods, not like just directly doing the surgery. And for ones that, it's actually affecting lots of one's brain and it's actually really serious, we can, use the surgery method. Thank you for the question.
Other questions?
Just curious how the fluorophores, especially in the brain, are actually excited to actually emit the light from fluorescence.
Because the fluorescent sodium is a type of dye, which after it gets into our sample, using fluorescence microscopy, we are able to, like, in a specific wavelength, we're able to illuminate the dyes, and therefore we're able to visualize the tumor.
Or comment, just so comment on your research proposal and say it's very interesting because you are bringing up like cutting edge technologies, especially like bringing the microscopy inside the surgical room, which is very, very innovative. So to answer your question, how do surgeons visualize the dyes, as you mentioned, by shining specific color on the surgical field? And it's really interesting because now the surgeon has to learn to operate with a set of goggles and kind of stuff, which is kind of science fiction, but it's actually happening. Congrats on your project. It's actually interesting.
Thank you.
Okay, thank you, group one. Okay, group two, come on up. All right, clicker to mobile and then this one as well.
Ready?
Yeah.
All right. Oh, sorry. Hello guys. My name is Ted Vaughn.
I'm Courtney Chang. I'm Kate Austin.
Today we're going to be talking about the use of cryo-electron microscopy for the study of piosins and how they can attack VRE.
Okay, to give you guys a little bit of an overview of what we're going to be talking about, in the medical world, we use antibiotics a lot to treat bacterial infections. And the way we do this is that we use antibiotics to kill bacteria. But nowadays, we're facing a lot of harmful pathogens that are actually resistant to antibiotics. So using antibiotics are not going to have as much as an effect. So for our presentation, we're going to be researching if we can use piosins to kill the harmful pathogen vancomycin-resistant enterococcus, or VRE. And it has a resistance to many types of antibiotics, and our research in general is hoping to stop antibiotic resistance. So we want to use cryogenic electron microscopy to look at the receptor binding proteins on the piosins to see how we can engineer them and change them so that they can bind and destroy the VRE. And then once we can prove that we can use the piosins to stop VRE, we can open up to the possibility and the idea of using these precision antibiotics to stop other pathogens and hopefully put an end to antibiotics resistance in the earth. So, as I said, we're using cryogenic electron microscopy, or cryo-EM, and this technique uses flash freezing of biological molecules in a solution, and it allows us to determine and analyze the structure of molecules and complexes that advance our research. And this was developed in the 1980s by Jacques Dubochet, Joachim Frank, and Richard Henderson. And hopefully our research is going to be helping the scientific field of bacteriology, which studies bacteria and its relation to medicine. Bacteriologists focus on the development and the growth of bacteria. And the pathogen that we're going to be talking about is VRE. It's resistant to a lot of powerful antibiotics like vancomycin and penicillin. And people who are at risk for infections because of VRE are people with lots of medical conditions or who have had many types of antibiotics in the past or who are chronically ill.
So what is a piosin and how does it work? So piosins are bacteriocides used by bacteria in order to kill competing bacteria. They do so by binding to surface proteins on the target cell and contracting, which will send a spike through the membrane and cause ions to flow out of the target cell, which will kill it. They consist of three major parts, which are the collar, which caps the piosin, the trunk, which has a sheath which contracts, and the base plate, which contains tail fibers. The tail fibers have receptor binding proteins that will bind to the ligands or surface proteins on the target cell. And when that happens, they will trigger the base plate to initiate contraction and kill the cell. So some previous research, a previous study done by Spiegelman and colleagues has already used X-ray diffraction to visualize the surface protein on the VRE, which we can use to engineer our piosin to bind to that surface protein in order to kill the cell. Other practices to reduce the risk of VRE have included hand washing practices, targeted surveillance cultures, isolation of VRE positive patients, and restriction of antibiotic use. But even with all of these attempts to reduce the risk, VRE continues to be a large threat to humans in places like hospitals where the bacteria attacks those who are already compromised by other ailments.
So to go a little bit more in depth into what cryo-EM is and how it works. So it starts out with our sample and EM grids, which are like tiny circular mesh made out of metal. So we put our sample and create a film of it on top of the EM grids, and then it undergoes plunge freezing. So we vitrify or freeze the EM grid, and this allows us to keep it in its natural state, as well as also aim to not have crystallization, and if we were to have crystals and crystallization, it would disrupt the structure of our sample. So after we plunge freeze the grids, we use an electron microscope, and we use this to image the sample, so we're gonna end up with multiple 2D projections of it, and then we're gonna use those projections to create a 3D model of our sample, and then we can use it to research and look at the surface and structure of what we want to look at. And then again, we're using it to look at the receptor binding proteins on the pads.
So our solution is this. We know VRE is resistant to lots of antibiotics, and we know that biosins can be used to eliminate this pathogen. So this will work by the biosins will kill the VRE by attaching to its surface proteins and penetrating the bacteria. The cell membrane barrier and the passing of ions across the barrier due to passive diffusion will destroy the cells. The use of biosins is far better than antibiotics because not only is VRE resistant to many of these antibiotics, but taking antibiotics to fight VRE causes further antibiotic resistance. Thus, as more powerful and last-ditch antibiotics are used to treat VRE, the bacterium develops further resistance and a dangerous cycle is formed that becomes harder and harder to deal with. Biosins are better at dealing with VRE because their protein receptor binding proteins can simply just be changed and adapted to match those of new VRE strains. There does not need to be huge sums of money put into going into research for cures, as proteins can be changed due to the nature and killing mechanisms of biosins. It is expensive to produce or discover new antibiotics, and it is far cheaper to just change the protein binding receptors of biosins. And future directions. Sorry. What is the best surface to put biosins on so that they're the most effective at killing VRE? Are they hospital beds? Is it countertops or medical instruments? Should biosins replace antibiotics completely in the battle against VRE? And can biosins replace antibiotics for all bacteria? And current future directions that extends the impact started by the turning point is, can biosins be made to effectively counter all other types of bacteria? Or could bacteria develop some type of resistance against biosins? And if so, how do we prevent it? And there's our references. Yeah, thank you.
Any questions from our audience?
So I have a question on how you're administering biosins. You mentioned putting it on like surfaces and medical instruments and whatnot. And I was wondering if that is more efficient than injecting like the biosins, if that's possible. Like I was wondering why you put it on like material surfaces as opposed to directly into like body patient who is at risk for infection and like I don't know, the bacteria.
So the thing with putting them on surfaces and medical instruments and countertops and whatever is that it's preventative. We don't want someone to get VRE and then have to treat them. It's like if we can prevent it before it happens. Furthermore, if we inject the biosins into someone, then it's a foreign object inside their body. It's your immune system. It's no different than a virus itself or like some like foreign pathogen or a bacteria. The immune system is going to attack the biosins. So the biosins will be attacked. So the biosins, they won't be nearly as effective, and they're going to make you sick. So it's almost like injecting like a it wouldn't be very helpful to inject it to the person. Nice question.
So my question is, so I really like the idea of using the biosins and engineering them to be able to target the specific bacteria. My question is, with cryo-EM, what information are you getting from it that will actually help you determine whether or not it's a good match? Like, what information would you get from that you couldn't get from just seeing if the bacteria gets destroyed by combining whatever pyogens you engineer with the bacteria? How is the cryo-EM giving you better information?
Well, the cryo can be used to study like the chemical structures and stuff and chemical bonds, and that could help us because we know the shape, and the shape could help us. So it's just more precise, and we can like tailor it to our needs, and we can make it faster at killing, or we can make it more precise, and you name it. Like there's lots of things we can do, but we can use the microscope to help us in that. Yeah.
We need some student questions.
Yeah,
see, okay, all right. Oh, good, good.
Me and a key class.
Stand up.
Okay.
Thank you, good presentation. Just to follow up on the last question a little bit, and you get the actual microscopy done, hopefully in 3D shape and results you get. I was just curious if you know if possibly what kind of modeling or AI is being applied to that. Because when you do change the proteins and receptors, you change the structure, possibly it's. So I was just curious how AI is being developed.
Yeah, that's a really good question. AI could be used to could be used to like test like algorithms and stuff, and then see which shape is best. Because like if we have like people do it and they could just test different things and do different things, it takes a long time, and it's a lot of just time to put into it and work and whatnot. But if we could just make AI do it, AI is like it's adaptive, it learns, and it could come out like in a much faster time and produce a shape that attaches to the protein. Yes.
Great job. Thank you, guys.
Okay, group 3. Let me just click on it to make sure it works. How do I click on it? Good question. Like this? Yeah, because right now the pointer's not. Oh, okay. Alright. Yeah, that's cool. Are you ready?
Ready.
No, but I'm gonna start anyways.
So, hi everyone. Today our presentation is going to be on the monitoring of retinal concentrations in blood, specifically the Accutane levels in pregnancy using fluorescent microscopy. So I'm Julianne.
I'm Elaine.
Kevin.
So starting with our abstract, the aim of this innovation is to allow pregnant individuals the choice to use Accutane, which is a isotretinoin, a substance that may cause birth defects if reached a certain intake level. We believe that everyone should be able to utilize this treatment, and we'll expand on why this is, as it is the most effective acne medication there is. If used properly, isotretinoin has around a 90% success rate, with the results persisting for life. Acne is the most common skin condition in the United States, affecting up to 50 million Americans annually. According to average results, it takes around 4.8 days to clear this medication from the system. However, this varies for each person. Therefore, we aim to develop an approach where individuals can monitor the concentration in their blood by themselves to utilize the medication safely.
All right, so I want to go over the basic concepts that will be used with our research proposal, right? So we will also be utilizing fluorescence microscopy similar to our lovely group 1. And Fluorescence microscopy is a technique used in with optical microscopy or light microscopy, where typically a laser will inject a specific wavelength of light onto a sample. But what's different about my fluorescence microscopy is that the sample will either have or be injected with fluorescent proteins or fluorophores. And another example of that is GFP or green fluorescence protein. And this will help them emit light at a certain frequency, which will help the sample or the area that is injected with the fluorophore in the state and able to be able to see more. The scientific field we're hoping that our proposal will help is the health field. So like we said in the abstract, regular doses of Accutane cannot be taken during pregnancy because it can cause drastic birth defects. But Accutane may be essential for women with bad acne or skin conditions. So our method could be used regularly to keep Accutane concentration in blood below a threshold that is definitely to the baby, which could have beneficials of the will keep the benefits that the Accutane will give to the women that have bad acne, but lower the effects of birth effects.
So how and why is using Accutane beneficial in our lives? As we know, acne worsens our facial appearance. Many people may suffer from insecurities and they get depressed. So using Accutane to reduce acne also can reduce emotional issues. So let me talk about the science behind it. Accutane is essentially a form of vitamin A. It reduces the size of secretion and sebaceous glands, which are the oil-producing glands in the skin, as shown in figure 1. So as we decrease the amount of sebum, which is the oil produced by the skin, we can thereby reduce one of the primary contributors to acne formation. Finally, we can also reduce the population of propionic acne on the skin, as shown in figure 2. This is microscope imaging. So this bacterium plays a significant role in the development of acne.
So I feel like Accutane is quite notorious for like causing birth defects in pregnancies, but I just want to further highlight with like statistics and data what exactly it is affecting. So the risk of Accutane is that there's approximately a 25% to 35% chance risk for the effects in infants exposed to the drug in the utero, and about 30% to 60% of children exposed to Accutane prenatally have been reported to show neurocognitive impairment. And this wavering in the percentage is caused by the study having suspects of Accutane exposure and like confirmed Accutane exposure in their pregnancy. And the dosage also has an effect on like the birth defects. And no association was found between moderate doses of Accutane and fetal malformation, but intakes exceeding 10,000 IU vitamin A significantly increases the risk of malformations. The World Health Organization recommends a dosage of 150 micrograms RA before pregnant women. And this figure, which one is the latest? Yeah. So this figure shows a fetus exposed to Accutane. And it has, you know, a very big forehead, fluid buildup in the brain. The ears are low set, and the jaws are also low set.
So what is our proposed solution? Basically, our microscopy technique will be fluorescent-based vertical flow assay. It's a paper-based fluorescent vertical assay network based quantification of biomarkers using cell phone. It is easy to use and is proven to be accurate, as showcased through a proposal that we were exposed to earlier this week. So it's accurate and inexpensive and easy to operate. It's better than lateral flow assays because it can produce 2D or 3D imaging. It can also detect very small concentrations. So how it works, it can ensure that the daily intake is below 2.5 micrograms, which is in the range of colorimetric and fluorescent microscopy. So it's around this range. So we will be using conjugated polymer nanoparticles, which is nanostructured materials that can be used in biosensing and nanomedicine, and is non-toxic. It is paper-based and therefore affordable. It is proven to be accurate, and this is very crucial for us because the intake threshold is essential to stay under. So if you can see in this figure, the concentration directly correlates with the fluorescent intensity, which is why it is very sensitive to any concentration changes.
So what are some future directions and questions you guys may pose on our project? What are some of the scientific evidence supporting this in order to make it reliable? How will informed consent be managed, ensuring that pregnant individuals fully understand the risks associated with Accutane use? How will this change affect public perception of drug safety during pregnancy? What changes in regulatory policies would be needed to allow this use, given the current restrictions under programs like the IPLEDG? And how will the health of the fetus and pregnant individuals be monitored during isotretinoin treatment?
Thank you so much.
Thank you.
Thank you.
Where's the questions?
Thanks for the presentation. Maybe I missed it. As a point of care device for personal monitoring, what bodily fluid would be measured when this would be eligible?
We would be using blood testing because it is already for individuals that are not pregnant, the conventional method to test the Accutane concentration in their blood would be through blood testing, right? Since this is a handheld device, you would be able to use it yourself. It's similar to like a diabetics test where you can like use it yourself with your own imaging device, such as a cell phone.
Will there be any difference in the effects? Like, currently Accutane has a large effect on depression and the emotions of people taking it, and pregnant women already have a lot of like that kind of thing happening. Will this new proposed solution differ in that aspect?
Right, so that's a really good question. Unfortunately, we were not able to research that aspect yet, but we would be able to later on, and it would better answer your question. So I would need to do deeper research for that. Thank you.
I'm curious if you guys were able to get to the point in your research where you were thinking about how frequently does a patient take Accutane, and at what point in that cycle of taking the medication would it be the most appropriate to get the most accurate measure of the amount that's in the blood, so you're not getting an abnormal spike high or an abnormal low, but that you're getting a good sort of average assessment of the level of Accutane.
Right, so this also depends on, we did consider this issue, but I feel like a lot of the concentration that you're taking, because Accutane concentrations can be varied, right? It depends a lot on the individual and the severity of their skin condition. So at this point in time, we are unable to give you a definitive response, but what we believe would be a good monitoring technique for this would to be like daily monitoring. So for like example, people with diabetes, they would have to monitor the glucose levels in their blood daily with that device, right? We want them to be able to get the benefits possible, but also like number one is their own personal safety. So we believe that frequent daily testing is probably recommended. I hope I answered your question.
You did, thank you.
Yeah, thank you.
Any last questions? Okay, thank you.
Thank you.
Okay, group 4. Guys, we are basically back on schedule, so we're killing it.
Hi everyone, my name is Michela Sherlatan and I go to Whitney High School.
I'm Ben McCullough and I go to PCHS.
And I'm Alexa Curtis and I go to St. John's School.
And today we're going to be talking about tagging amyloid beta proteins in primates with Alzheimer's disease using GFPs or green fluorescent proteins. So to give you a little overview of what this is about, amyloid beta proteins are correlated with Alzheimer's disease and often form plaque in the brain that contributes to neurodegeneration. Using GFPs to tag A beta proteins is a developing approach to studying AD in humans. We can do this by using GFPs to track and measure the amount of A beta proteins in primate brains, which will reveal either a positive or negative correlation between disease progression and the amount of proteins. Having a strong understanding of these proteins will allow scientists to improve and develop new treatment options for patients and explore correlations between Alzheimer's and other neurological diseases. So the microscopy technique that our idea utilizes is fluorescence microscopy. And in fluorescence microscopy, scientists use high-intensity light to excite fluorophores, which then emit light to allow for high contrast imaging of certain biomolecules, which in our case would be the A beta proteins. We then use GFPs to tag these proteins to get a clear view of the pathogen concentration of the proteins in the brain. The scientific field that this idea would help give insight to is the field of genetic neurological disorders, more specifically Alzheimer's disease. The research can give us information that can help scientists create new treatment options and eventually find a cure.
Scientists are currently using a number of techniques to research Alzheimer's disease: animal studies, head scans, genetics, and clinical trials. For animal studies, animals are given Alzheimer's-like pathology where the disease is researched while potential treatments are tested. For head scans, these consist of PET scans, MRI, and CT, which overall are just used to study and visualize the brain, and in the case of PET scans, detect any abnormal protein buildup. In terms of genetics, there is genome-wide association studies, or GWAS, which have been used so far to identify genes associated with Alzheimer's and even label them as risk factors for carrying the disease. However, there is still a lot to learn in terms of the specific role these genes have in the disease's progression. And this picture here shows of over 2,000 clinical trials, the various hypotheses that have been tested. So the amyloid beta, which we're looking at, is only like 22%. This just shows you how divided the scientific community is on this topic. And we're using fluorescence microscopy, which uses dyes, in our case, green fluorescent protein, attached to specific structures to visualize them for better observations. This experiment specifically uses STED, which is stimulated emission depletion, a type of fluorescence microscopy with 2 lasers to create high-resolution images.
As scientists continue to explore the connection between the amyloid beta protein and the development of Alzheimer's disease, 2 diverging ideas emerged. One study says that AD is caused by a lack of beta amyloid proteins, while another claims that Alzheimer's is caused by buildup. In order to determine which standpoint more accurately describes the effects of amyloid beta on Alzheimer's disease, we came up with this proposition using primates. So our proposition is that amyloid beta proteins inside of primates genetically modified to have Alzheimer's disease will be observed at certain points in their progression of Alzheimer's. This enables scientists to see how the amount of amyloid beta proteins contributes to the development of Alzheimer's. So to do this, we will first use CRISPR, a gene editing tool, to mutate the amyloid precursor protein, or APP gene, to the known mutation for Alzheimer's in a monkey. Then we will attach green fluorescent proteins to amyloid beta proteins by fusing the ends of the APP gene to the codons for the GFPs. At different stages of development, we will deeply anesthetize a select group of monkeys and basically freeze their bodies and then take samples from their brains later on. These brain samples will then be imaged using a stimulated emission depletion microscope, or STED, in order to reveal where the amyloid beta proteins are. So why is our proposition better?
Well, we have two reasons. One is that primates are useful when looking into diseases that develop with age, such as Alzheimer's. This is because their brain behavior emulates that of humans. And two, fluorescence microscopy, specifically STED, enables us to see and approximate the amount of AB proteins present at various stages of Alzheimer's disease. Finally, for our future directions, after this experiment is completed, we have two emerging questions that we found to be the most important. The first question is that, does the amount of amyloid beta proteins correlate with the onset of Alzheimer's disease in non-human primates? And then after that, what about humans? And our second question was, how will fluorescence microscopy continue to help understanding AD and other neurological diseases? To move towards answering these questions, various extensions are possible. For example, future experimentation involving neurological diseases in fluorescence microscopy includes imaging the other protein correlated with Alzheimer's called the tau protein. Tau aggregates in certain locations in the brain in various diseases, including Alzheimer's. Although the effects of a mutated tau-like gene have been observed, fluorescence microscopy will enable correlations between tau proteins in Alzheimer's versus other neurological diseases such as Parkinson's to be evident. And thank you.
Questions in our audience, please.
Two questions, I guess. One is, why do you need super resolution optics for this measurement? If you're not particularly interested in structure, you're looking at concentrations of fluorescent protein in a tissue. And then two, did you think about any other alternative solutions that don't have to basically kill primates to do this work? Did you look into anything where there was microscopic tools available for monitoring behavior in freely behaving animals? I don't know if it's been done with primates, but it certainly has with mice.
Yeah, we struggled to find a separate technique because primates are like much larger than mice. So we didn't really find a way to like be able to take brain samples from a primate or like scan the brain without like killing them. And what was your first question again?
Why do you need super resolution optics for this? I mean, it's obviously a powerful technique, but if you're not necessarily trying to understand the structural properties, you're just detecting the presence and then ideally mapping that onto the concentration of GFP. Do you need super resolution optics? Because that complicates things quite substantially.
Yeah, we were concerned that because in Alzheimer's disease, amyloid beta proteins, they usually build plaques between neurons inside the brain. So we're concerned that that maybe would like inhibit us from fully like counting like or like recording the amount of proteins very well.
Other questions?
Great talk. So if you have to give a suggestion why this plus problematic, based on your reading, what would you say?
These plaques, in Alzheimer's, they found that a lot more plaques build up in the brain as like a human or a primate would age. They found that, not exactly sure, but these plaques, they will destroy the neurons attached to it, which would make it hard for us to like see like how the proteins themselves, like how the quantity of the proteins affects it rather than the plaques that they build.
Interesting. I think that I agree with you. I think that's a whole problem.
So if you were to do these experiments, and I would like to put it in the context of what we do as scientists, because we are concerned still, using monkeys, for example, we try not to do everything we can to replace its higher vertebrates with other models. So during the class, we spoke about a lot about other models that can be used. Speaking monkeys, for example. So, like from what you read, have you come across other models that are being used to study Alzheimer that can actually mimic what's happening in the brain of that can be used instead?
Yes, we did find several studies using mice, but we just thought it would be better to use primates because they are closely related to humans. Even though it is harder.
What about zebrafish, for example? Have you read about that?
Not for Alzheimer's because like the main problem of Alzheimer's is that it's onset with age. And like monkeys like and mice, they closely like emulate how like the human brain ages, whereas like zebrafish, I don't believe they quite age in the same way. Or yeah.
Okay, great job, guys.
Okay, group 5.
What's that drive tab that's on the front right at this point? Because I think that was just another big one. Yeah.
Hi everyone, so today we're going to be talking about transmission electron microscopy applied towards analyzing extraterrestrial samples of ribose on meteorite rocks. So my name's Stephen.
Hi, my name is Celia.
Hello, my name is Sean.
So the research question that we're asking ourselves is how can we identify the beginning of extraterrestrial life through meteorites by utilizing microscopy? So to achieve that, we're going to use transmission electron microscopy in order to sample meteorites and search for ribose. So ribose is a type of sugar and it's one of the fundamental building blocks of life. If we do find ribose on the meteorites, so there would be a high probability that it could sustain life on extraterrestrial planets. So previously attempts to locate ribose have been successful, but they do use non-microscopy methods and they're tedious and long. So in order to solve that, we could utilize transmission electron microscopy to streamline this process and sample the meteorites towards the search of extraterrestrial life. So our concept and topic, so the microscopy technique that we aim to use is transmission electron microscopy. It's because we have to slice our sample into very thin pieces in order to find the ribose within the meteorite. And transmission electron microscopy provides us with that advantage. And the scientific field that we're aiming to further research in is astrobiology. And that's because we're aiming to find evidence in the extraterrestrial domain.
So the precedents that we took for our final involve, before our proposed solution came along, previous astrobiological endeavors of finding ribose on meteors were by international teams of NASA, and they were also majority of them were successes. And said international teams would utilize hydraulic acid and water from the meteorites to extract and liberate all the sugars, which are essentially the ribose from the mineral surfaces, and continue to purify and chemically alter them. Additionally, other endeavors such as those such as the ones that I've been talking about included the use of a gas chromatography, mass spectrometry, which is a process in which molecules are sorted and identified by their electric charge and mass. So we're using transmission electron microscopy specifically because of its advantages. So particularly how that works is how the microscope for it works is that a beam of electrons is essentially transmitted through the sample or the specimen, and which creates an image. And that image is basically formed through the interactions of that beam of electrons and the sample. What's specifically notable about this microscopy is that it uses it particularly has a higher resolution than most light microscopes, so that's why we think using TEM would be more preferred, more beneficial for this endeavor. And of course, and TEM has been previously utilized to make observations and determinations during RONC investigations and observations, experiments, and all of that. And according to Leisha Microsystems, that a professional source. Ultramicrotomy is a process most associated to TEM. Is a process in which materials are thinly sectioned for microscopy, and it facilitates a proper analyzation and visualization of the structures of the samples.
So, yeah, so basically we just continue the groundbreaking work of the laboratory that discovered ribose in meteorites. So meteorites contain ribose, and so it supports the chemical preparative theory. So this theory actually suggests that the elements necessary for life may have been delivered to Earth by meteorites or other extraterrestrial rocks. And so now we know that meteorites contain ribose, and so actually ribose is an essential component for life. So it's a really huge finding. And so, as he said, we just propose to examine and to see these ribose more in detail. So it's TM, so transmission electron microscopy. And it will just allow us to observe the structure and properties of the ribose more closely. So, so we are going to use TM. So TM, there's like a tricky thing is that the samples will be really tiny, between 50 to 100 nanometers. And so the sample preparation is going to be really ridiculous because we are actually, we study rocks, and so it's really solid. And so it needs, the ensemble needs to be really tiny. So it's going to be really hard to do the sample, but we're going to use several elements. So first we're going to use diamond or so to just cut into our meteorites, because actually it's really important to have the inside of our meteorites, because when the meteorite arrives on Earth, its surface is completely destroyed when it crosses the atmosphere. So all the potential organic molecules that found on the surface will be destroyed. So after we're going to use electromechanical pollution to just clean the surface to have a really clear TM information. After we are going to use microtome to just select the area that is interesting, and after we're going to just put the sample layer of gold. So basically, when the electrons incident electrons emitted by the source particles sample together internal information, and from the main image, there are other electrons that are affected by the gold, and it will provide us surface information to have a better contrast image and have just a better image to see our ribose.
So our future directions, our next steps. So after discovering ribose on a meteorite sample, we will understand what life forms could be supported from the meteorite's origin. And then we further the research for bacteria and other essential building blocks of life to know about the types of life that can be supported in space.
Yeah, so here are all of our references that we used. And thank you for listening.
Yes, thank you for listening. Oh, yes, questions, if anybody has any?
Person, microscopy methods, is the issue that, the concentrations are super small. And then this overcomes that concern, I wanted to understand, other ways of detecting ribose and how this is better.
That's a very great question. So, yeah, I mean, we just originally thought that TEM would be better because of its there's more advantages compared to other forms of microscopy. So while let's see.
Okay, so previously they didn't they did detect ribose on meteorite samples, but they didn't use a microscopy technique, and I think they used some kind of acid to dissolve the meteorite, and they detected the concentration of ribose within there. So we felt that it would be much easier and much faster just to use the microscopy technique, where we just transmission electron microscopy, where we just create thin slices and then we're able to image that.
And so, actually, when they did their experiments, it was more for, they focused only on like chemical and scientific analysis and not on morphology of the ribose. And so TM, with TM, we can actually really see completely our ribose, and it's like a completely different experience. Like we don't see this, we don't try to see the same thing on ribose.
Very nice. Exciting to look at this question about the electron microscopy that we use. You heard from the lecture that electrons very damage, and look at it, destroy things. So if you worry about it, what is a potential solution that can alleviate the problem damage of your sample?
Yeah, I think it's complicated because in fact meteorites are really rare, so it's a huge problem because it's not like random rock on the earth, and so it's really like we don't have a lot of meteorites. So I think that it would be I don't think there is a solution for that, but we will just like analyze one really tiny sample of, for example, the Murchison meteorites that is really famous for, because we just found a lot of organic molecules on these meteorites. But I think that it will just be complicated, but we will just like analyze a really tiny sample of the meteorite. But I don't find another solution for that.
Is there another sample preparation technique before you use electron microscopy that would help preserve your sample? Manifest it hand motion.
So yeah, there could be other methods that could be used, such as cryo-electron microscopy as one, to cool, as I'm sure you've all heard of, putting samples into cool cryogenic temperatures. That can also be another option we could definitely utilize. So
not so much a question, but just more a general point for you and for everybody, right? One of the things that you should walk away with this week from is an understanding or appreciation for that. Every microscope has an advantage and a disadvantage. It has limits and it has possibilities, right? And so while we talk about beautiful images of atomic resolution, right, whether it's with the electron microscope or the scanning probe microscope, pushing the limits of speed and space and time with optical microscopes, it doesn't work for everything, right? And so, for example, I was just asking, ribose, yes, we can see atomic resolution structures of biological materials, right? But ribose in particular, it's not unique, but it's very small, right? And so to see something that small with that type of resolution generally requires a crystal. right? And I know you didn't learn about this technique, but there's a new sort of an extension of electron microscopy called microelectron diffraction that sort of takes traditional X-ray diffraction and uses electrons. That is the solution that I would presume I would go after, right? To try to get the structure and identification of a molecule that's that small. Right, that's why a lot of the stuff you saw in cryo-EM is viruses and proteins. They're bigger, hundreds of kilodaltons and larger. When you start to get smaller, right, you have to think differently about things. The same thing is true with the other microscopes. And so for all the projects, when you leave here, right, like we keep telling you about all the great things you can do, but everything has, there are boundaries about what's possible with the current technologies. And so really thinking about your sample, how you're going to prepare your sample, right, and what you can actually utilize, because it doesn't scale infinite. I showed you images yesterday at atomic resolution. You can't get atomic resolution on everything, right? Just because you can get it on something.
Because we need to like choose just one microscope, just chose this one, but of course I think that every science research you need of course to like use multiple microscopes to see different things. So we are just focusing on TM, but like
no, TEM is the way to go. I didn't mean to say that it's not. Right?
Yeah,
it's just again, it was more just a comment for everybody. I think it's just a reminder.
Okay.
We're exposed to so many amazing images, right? But everything has its specific considerations that need to be taken into account. But TEM, for what you're proposing to do, is the solution.
Yeah.
There's just a slightly different version of it that you didn't even hear about.
Yeah, all right. Okay.
All right, thank you guys. Thank you very much.
And just like that, we are back on schedule. Woo! Okay, so we are gonna take a 10-minute intermission here. So we're gonna be back here at 11:30. Okay?
I'm gonna pause the recording.
Okay, we're gonna get started again. I already restarted the recording, Rita, so we're good on that end. I've unmuted myself, so everyone should be able to hear us again. And then this way. Oops. Group 6. And I'm ready.
I'm Mackenzie.
I'm Alice.
And I'm Julia.
And this is our presentation on Can Vision Be Changed? And here are the contents of our presentation and the 10 slides that will be presented.
So to give you an overview, our objective is to observe the change in the DNA structure and the gene expression that occurs when a nocturnal gene is being combined in an embryo of the diurnal animal gene using transmission electron microscopy. And from that, our goal is to produce an animal with both nocturnal and diurnal vision, and as well as determine if the modified animal can see at all times of the day and night. And by doing so, it will be feasible for us to see the cellular interactions occurring within the rods, as well as how the organisms, photoreceptor cells work and adjust to different light conditions.
So this is the concept topic slide. The microscopy area we are presenting is TEM, transmission electron microscopy. We would use it to compare and contrast images of photoreceptor rod cells of nocturnal, diurnal, and hybrid mice. TEM uses a beam of high-energy electrons to shine through the sample of rod cells onto a detector to get the image. The scientific field that we are helping with TEM is the field of optics and of genetics. In a way, we are combining these fields by using a genetics technique to study the science behind vision. Precedence Part 1. Our solution involves injecting DNA from the rod cells of the diurnal mammal into the embryo of a developing nocturnal mammal such as a mouse in an attempt to give it a combination of diurnal and nocturnal vision. Although this has not been done before, there has been a study where nanoparticles were injected into an adult mouse to make it able to see infrared light. Another study showed injecting external DNA into a mouse embryo to change it. Researching this idea could revolutionize the fields of optics and of genetics.
And the microscopy technique we're using is a type of electron microscopy called TEM, also known as transmission electron microscopy. And it's a form of electron microscopy in which an electron beam is transmitted through the specimen to derive a 2D image. But similar to a CT scan, we can combine many of these 2D images to reconstruct a 3D figure, which is a process known as 3D reconstruction. And since the beam of electrons is penetrating through the specimen, the sample must be very thin. And the main benefit of using TM is being able to see what's inside the specimen rather than what's on the surface. And since we want to monitor the injected DNA inside the nucleus over a period of time, we're using TM. And the top image for figure 2 shows an image from a TEM from a microscope, and the bottom is an SEM from an SEM, which is a different type of electron microscopy. But the reason why we chose TEM is because we want to see what's inside the nucleus, so inside the cell, rather than the topography or the surface of cells. And here's our solution. This is our proposal. So our research is about attempting to create an animal that has a combination of both nocturnal and diurnal vision. And we will be monitoring the weekly development of the rod cells of the hybrid animal in order to observe the change of the proposed gene expression. And to do this, we will inject DNA of the rod cells of a diurnal animal into the nucleus of a mouse embryo. And then we will image the rod cells of the hybrid mice they develop using the TEM. And this way we can compare the image of the hybrid mouse cells with an image of a normal mice rod cells. And the figure on the left shows the development of a mouse's rod cells.
So our method is that we're gonna micro-injecting the DNA using fine glass micropipette from a diurnal animal into the nucleus of an embryo mouse, but we need to ensure that the embryo stays alive. So as you can see in figure 1 is like how we're gonna do it. So next, we're gonna monitor the photoreceptor cells of a baby mice once a month by taking a small sample of the retinal rod photoreceptors from the eye of the mice, and we will use PCR to ensure that the injected gene is visible. And then we would examine whether the hybrid photoreceptor cells have characteristics of both nocturnal and diurnal vision. And at the end, we will utilize TEM to view and examine the sample to see how the DNA has integrated into the nucleus, as well as how the mouse cellular structure.
Our solution, why is our idea better? A previous study researched how rod photoreceptor cells in nocturnal mammals differ from those in diurnal mammals or day animals. We discovered that a nocturnal mammal's rod cells are more efficient at collecting light than those of a diurnal mammal. However, that study did not attempt to combine nocturnal and diurnal rod cells together in one organism, where our research would find the answer to that question. Our work does, however, still have its limits. For example, attempting to modify an embryo and having to constantly monitor the development of the injected DNA makes it a difficult process. Since there is also limited background information on this topic, it is difficult to find suitable methods for our research, and we are unsure if it will produce our desired result.
And now for the future directions and possible questions. So first is, can a single animal even utilize both nocturnal and diurnal vision? What will the benefits of having both be? And what could be any possible side effects of injecting DNA into a developing animal and embryo?
And after our investigation, we can keep track on the long-term impacts of genetic mutations of the modified mice behavior and also their ability to reproduce. And we can also investigate on how their brain can accommodate both eyesight by thoroughly examining on how the mice or like another animal we might investigate later interprets signals from both diurnal and nocturnal light receptors.
And here are references and the contributions. Okay, questions from our audience, please.
Really interesting idea. I'm curious, sorry if you addressed this and I missed it, but can you clarify what specific type of information you're trying to get by using TEM specifically? And I guess sort of as an add-on or a part two to that, are there any other techniques that you heard about this week that could give you maybe even some additional information beyond what TEM could give you?
That's a really great question, and basically the reason why we decided to use like TM is because oh, sorry, for your first question, were you asking, was that what you're?
Yeah.
Okay, so we were using TM because we want to see like the development of the rod cells and to see like if throughout the development, if the hybrid rod cells will combine aspects of both like a diurnal, like DNA from a diurnal animal and DNA from a nocturnal animal. So by like monitoring it like monthly or however often we were gonna do it, we wanted to see like if it will have like aspects of both, or will it maybe develop like another like characteristic.
So like morphology aspects, physical, like what aspects do you think are key here that TEM will tell you?
So there's actually like an image that we had. Did you have it here? Yeah, I think you still did. Oh. There it is. This. Sure. So this is the image of a mouse's rod cells versus like a diurnal mammal's rod cells. And TEM helped them like see like what the difference is. And like the mouse's rod cells like help it collect more light. So we would use TEM basically like to get an image like this to see like what aspects are combined, like how they look. Like continuing off of that, as you can see, like the top image of a like nocturnal animal, there's like one big blue like circle in the middle, and then on the outside there's red, and on the outside of that there's green, but then on the bottom image there's like multiple circles of the blue and red, and then there's like more like green in the middle. So I guess like what we're trying to like see is if like the hybrid animal cells will look like the top one or the bottom one, or like a mixture of both. But since like the characteristics of both are like so different, that's kind of like what we were looking to see.
And there are many differences between nocturnal mammals and diurnal mammals besides their rod cells, right? And so when you're thinking about looking at behavioral outcomes, like how maybe because of the hybrid animals, they behave differently from the original animal. I guess you're separating out the part that's just due to rod cells. What behavioral changes could you expect to see? What are you thinking?
So that's a really great question, but I think we would probably have to do like some further research, since we like specified our research on just like eyesight, and we didn't really get a chance to look at like behavioral differences. But that's a really good question, because with like combining like DNA from like different animals, you would probably like expect like something else to like happen as well. But I think we would probably have to do like more investigation on that to give you like a final answer.
Any other questions? Okay, thank you, Group 6. Okay, Group 7.
Hi, my name is Lexi.
I'm Peyton.
I'm Ulysses.
And our proposal is enhancing cancer detection with the use of fluorescence microscopy.
This is our synopsis. If you'd like, we can go back later, but I think we're just gonna dive right into our concept specifically. So our concept is we're gonna use fluorescence microscopy combined with fluorescent tagging techniques, and then we're gonna do a very detailed neuronal structure of the brain, primarily targeting like the function of the brain, but also to lead into better access to diagnosing cancers. We believe this microscopy technique is very useful in the medical field and specifically in diagnosing cancers, and cancer research could benefit significantly from fluorescence microscopy because it enables the detection of cancer cells. It's very detailed. It's very detailed, and also it provides a very good image of tumors and morphology.
Okay. In current state of science, fluorescence microscopy is used to highlight the surrounding of the cell instead of the cell itself. This is negative contrast fluorescence imaging as seen in the top figure, whereas we want to image or highlight the cell itself. Some studies of neural activities use genetically encoded indicator proteins that modulate their fluorescence signals in response to chemical or electrical events in the brain. And traditional cancer diagnostics rely on imaging techniques such as the MRI and CT scans, which may lack sensitivity for detecting small or residual tumors.
A previous study, like very similar to what we plan to do in 1999, Russian scientists were able to isolate the red fluorescent protein. And with this discovery, they used the colors cyan, yellow, and red to basically color a mouse's brain. And this technique drove the cell to switch on fluorescent protein genes. And they basically, as you can see there, they colored the mouse's brain, which provide just a very detailed image of the brain. So it's kind of similar to what we plan to do.
So what our solution basically entails is using fluorescent tagging, and what we are going to do is to tag cancer cells with fluorescent proteins or dyes that bind specifically to the cancer markers. And we will be using STED microscopy to visualize and also analyze the tagged cancer cells with a high resolution, which I'll get onto later. And finally, in the monitoring, we will basically be regularly image the patient to detect residual or recurring cancer cells early on in the stage. And the principle, basically, what is STED microscopy is, so you might be wondering, what does STED stand for? It means stimulated emission depletion microscopy. And so how it works is basically, the first you can see in the graph over there, at first by exciting a sample of the conventional laser. Now that laser for the wavelength is around 250 nanometers. However, we want to get that small as possible because we want to have basically a very detailed, a more detailed with a higher resolution. So what STED basically does is we have a little depletion laser that is kind of like a donut shape, as you can see in the middle. So what we end up with is around 50 nanometers of emissions right in the middle. So it's very, very small laser resulting in a very clear and high resolution image.
So some of the advantages of our proposal is that our technique is highly sensitive and will be able to detect low abundant cancer cells that may be missed by other imaging techniques. And we will identify recurrent cancer at an early stage, improving treatment outcomes. And it will target specific cancer markers, reducing false positives and negatives. And the main reason why we chose this microscopy technique is because it's non-damaging. Fluorescence microscopy is known to not harm the sample.
So basically in our experiment, we're going to inject the cancer cells like into the brain of a mouse. The brain of a mouse kind of emulates the human brain, so that's why we chose a mouse, and we're gonna inject it through the cranium, the bone that protects the brain. Then we will proceed to inject the GFP protein, and this protein will latch onto the cancer cells in the brain, and it will detect the tumor. Our experiment, we will know if it's successful, if we are able to successfully detect the tumor at an earlier stage than previous techniques are currently able to.
So one of the examples that we are going to use is pancreatic cancer. So why pancreatic cancer? Because pancreatic cancer is actually one of the cancers with the lowest survival rate. The early stage of cancer, it has a 5-year survival rate around over 80%. However, once the cancer has spread beyond these areas known as metastatic cancer, the 5-year survival rate plummets down to 2.4 to 3%. So that's why it's so important to detect these cancer early on in the stage, so we have basically time to treat the cancer. So how will we administer this? Well, as you can see, there's a big needle with fluorescence. We'll basically administer fluorophores, such as 5-aminolevulinic acid, which is a type of fluorescence, as well as other tumor selective agents that will specifically bind to the tumor, which accumulates in cancerous tissues. And how it works is certain molecules are optimally overexpressed or underexpressed in specific types of cancers. For example, the MUC1 gene is overexpressed in 90% of human breast cancers. So once we basically detect that gene, scientists can most likely accurately predict that, hey, this person might have breast cancer early on in the stage, and this can be like a target for fluorescence. And also, second, how it works is that, as you can see on the little picture there, the tumor tissue shows a stronger red fluorescence compared to normal fluorescence rates into normal fluorescence. So by using this, we can easily tell if someone has a tumor or has cancer or not. And then these are just some information about fluorescent emissions extends from 250 to 700 nanometers of wavelength. And then we can kind of compare this to a healthy tissue.
Some future directions for our project, we expect questions about if the actual protein will be reflected externally on the human brain. In previous examples using this protein, mice and other animals have actually turned the green color, but this actually probably wouldn't, well, it definitely wouldn't happen with the brain due to the very small amount of actual GFP protein being used. It will not reflect externally onto a human. In regards to the future directions of our project, we think that this technology would be very useful in many different kinds of cancer, specifically breast cancer and skin cancer, I think could be positively affected from this technology.
These are our references and then contributions.
Thank you.
Thank you.
Questions from the audience, please. Like when you're doing the earlier detection, can this help target treatment or identify the area for treatment, or does it have to be used with other? Like if you detect it early, how does it change the course of behavior?
So our main course is first detection early, because if we detect it early enough, we will have a higher survival rate for the patient. But also we try to make it as non-invasive also as possible, because a lot of the techniques right now, such as X-rays or prostate exams, it's very, very invasive. However, we try to make it less invasive as possible instead of using the X-ray.
Does this method enable you to detect the area that?
Yeah, the fluorescent. Yes, yeah.
It'll actually target the protein that is I think we had a slide about it, but we basically it'll target the protein with the cancer, and having it being detected earlier on, cancers tend to spread throughout their area, and we want to try to avoid that because that makes them more risky for them.
Yeah, it tags onto the cancerous cells and also shows a different kind of fluorescent light other than healthy tissues. So we can be able to tell that apart from, malicious cancer cells with normal cancer cells.
It'll be like a neon green highlight on the specific cancerous protein.
Thanks for the presentation. So the STED microscope you saw here, a couple of them, I think, had a very specific form factor. The microscope objectives are separated from the sample by hardly no space. How do you imagine translating super resolution microscopy onto a patient?
Right? You're gonna be injecting it into a living organism that is walking around. Mine, yours, head is not gonna fit into that microscope. Are you gonna be developing a new microscope solution that leverages STED? And then second question, I believe it follows on to one of the earlier presentations. Getting the light through an opaque sample is not trivial. So I think you saw the multiphoton microscope as well that allows you to do deep tissue imaging, like get further in there, as opposed to just like the superficial layers at the top. Did you think about the challenges of trying to actually image deeper into the tissue?
Yeah, so for the first question, that would work in a biopsy if we were going to basically perform surgery and basically take a tissue or like like a tissue out of the patient, then we would be able to use it. And then I'm sorry, is that your follow-up question? Does that answer your follow-up question as well? Because we're not obviously.
It depends on how big the
Yeah.
biopsy is.
Yeah, obviously, yeah. So
If it's a layer of cells thick, then you don't need to worry. But if you have even a microscopic volume of tissue.
Yeah.
Light doesn't go through opaque things, right? Unless you play some tricks.
Yeah.
And deep tissue imaging with super resolution is exciting, but it's not. Did they talk about clarified tissue? Right, like clearing tissue. So those sorts of things. There's just myriad challenges.
Yeah.
Right, to the approach.
I think for now, to answer that question is using like a biopsy, because obviously we're not going to fit the whole patient under the microscope. But obviously with the deep tissue question, using that microscope, we haven't obviously had enough time to done enough research into other microscope techniques or other techniques. We've basically mainly been focusing on this specific technique for this presentation.
Any last questions? Okay, thank you guys.
Thank you.
Okay. Group 8.
All right, so our presentation is on the field of confocal microscopy and its applications in the field of optometry. I'm Lyra Cunningham. I'm from Carlmont High School.
I'm Yi from Temple City High School.
I'm Lucas Chu from Cancast Academy.
All right. Okay, so abstract. Our focus is going to be on managing the progression of corneal dystrophy through advanced microscopy techniques and by combining in vivo confocal microscopy and fluorescence staining. So corneal dystrophy is a genetic eye disorder characterized by abnormal material accumulation in the cornea leading to vision impairment. Traditional methods often fall short in providing detailed layer-specific analysis, and by using a combination of in vivo confocal microscopy and fluorescence staining, we can achieve high-resolution in vivo imaging of the corneal layers, highlighting defects and anterior film status. So this basically allows for precise monitoring of disease progression, and a combination of techniques allows for detailed layer-specific analysis, facilitating better management of the disease and improving treatment strategies, ultimately enhancing patient outcomes. So let's take a look at the microscopy concepts that we'll be using for our proposal. So we are going to be combining in vivo confocal microscopy, or IVCM, which basically involves a laser that scans the cornea, and the reflected light is used to create detailed images of the corneal structure. And this technique allows for high resolution imaging of the corneal layers at a cellular level. It's not invasive and can be performed on the living eye. And when combined with fluorescence staining, which is an orange dye that highlights areas of the cornea when illuminated with blue light, it helps in detecting corneal injuries or diseases and can be used safely on the living eye and Some basics of that is when the corneal surface is normal, it appears uniformly green. And if there's a defect, the green dye will adhere and stand out. And so by combining both of these, we're able to detect issues in the corneal layer and then zoom in on them using the in vivo confocal microscopy.
Okay, so basically right here is just a picture of what in vivo confocal microscope is. And so basically what is better is just like it uses everything that a regular confocal microscope is, but what's better is that it's able to like view a live specimen, so we don't have to take a dead person's eye out, and it uses a less powerful laser so it doesn't damage the eye.
Here's an image of the fluorescent. Oh, right. Yeah. Here's an image of the fluorescent staining on the eye. Like as you can see here on the cornea layer, that's the fluorescent like adhering to the damages of the corneal dystrophy. You can see the patterns. Normally, like you would see like just if there was like no like problems with the eye, like if the patient doesn't have corneal dystrophy, you wouldn't see this kind of pattern on the eye.
Okay, so we should go over what optometry is. So basically, optometry is like the branch of medicine that deals with the eyes and visual systems. And optometrists are the professional healthcare workers that care for your vision, such as sight testing, diagnosis, and managing your vision changes. And so the current methods of diagnosis is basically just to diagnose corneal dystrophy involves usually routine eye exams and using slit lamps. And the corneal dystrophy has a few common symptoms like blurred visions, sensitivity to light, or double vision. But most commonly, it's just there's no symptoms at all. So it's like possible to know how you get it. And usually the only types of reliefs that the doctors could give the patients were lubricating eye drops or corrective lenses, but sometimes there were only temporary fixes because it came back. And optometrists had to rely heavily on patients to self-reports on examinations from a visual acuity test, which is like sharpness of your eye, like testing, like if you can see like these letters or not, and routine slit lamp exams.
Okay, and so let's look at how we're going to improve the current state of the topic. So by using a combination of confocal microscopy and fluorescent staining, we're able to get high resolution 3D images, in vivo imaging, which means we don't have to take out like a biopsy. This is connected to the person, and a diagnosis of corneal dystrophies and infections. And then this helps evaluate surgical outcomes. So what we're doing here is we're not necessarily proposing an idea for, you know, when to like how helping surgery, and more so managing the progression of the disease and understanding when to step in. And the fluorescent staining highlights the corneal defects, assesses tear film, and is non-invasive and quick. It diagnoses abrasions, ulcers, and dry eye disease.
You're proposing a more accurate and efficient way of monitoring the progression of optic diseases and disorders such as corneal dystrophy and experiment to test it. By using IVCM, we can observe eye disorders like corneal dystrophy without damaging one's eyes as other microscopy techniques like electron microscopes or AFM. However, one can complement this microscopy technique and include fluorescein to increase efficiency further. As shown in a previous slide, the ability to observe discrepancies in the eye becomes much easier when the eye is stained with fluorescein. And by breeding mice that have the autosomal recessive, autosomal, I think it's dominant disorder, an accurate experiment becomes much more feasible as then. The supply for mice with corneal dystrophy will not be a problem, meaning more rounds of experimentation can be conducted for precision.
So how does it work? We're using a combination, again, high resolution imaging is possible when you combine confocal microscopy and fluorescent staining by understanding where to get the images. And the layer specific analysis is crucial to manage the progression of corneal dystrophy.
So why is it better? IVCM with additional fluorescence comes with many benefits. For example, diagnostic accuracy. IVCM is able to investigate numerous corneal diseases by capturing high resolution microscopic images. Precision. IVCM allows for detailed visualization of cellular changes in corneal layers, aiding in wound assessment and diseases monitoring more effectively. Early detection. IVCM's capabilities enable fast identification of corneal dystrophy by examining the microscopic structures and examining abnormalities.
So the discussion our project hopes to raise is that just like corneal dystrophy, there are other eye diseases that have severe complications. For example, glaucoma is another eye problem that requires the same like comprehensive testing to like be diagnosed, but its symptoms are much worse. And in the future, we could potentially apply our IVCM technique to other disorders and diseases. And this project also aims to explore the potential management of these problems by using IVCM and fluorescence staining in the future. So contribution.
I'll reference it. And then thank you for listening and you don't have any questions.
I just wanted to get some clarity. Is the suggestion for normal people going to the optometrist to universally get these tests because they're low cost and low friction with patients? Or is it only when they're diagnosed with eye diseases that they'll have this test done?
So this is managing the progression of the disease. So typically, it's difficult to diagnose, but it gets worse with time. And so as you age, as it gets worse, the corneal will dystrophy more. And so this determines the rate of how it's like dystrophying. And then it helps determine when more invasive clinical procedures like corneal transplants need to be operated on the patient just based on these low cost testing.
Good presentation. I probably missed this part, but how are you going to dye the eye with fluorescence?
It's pretty easy. You get like an eyedropper and it's like non-invasive, it doesn't hurt the eye, and it's just a stain, fluorescent stain that as you can see, I don't remember the really kind of gross looking picture of the blue eyeball. That's just where you get the dye, you drop it on the eye, and then you use a blue light and you shine it on there, and wherever the abrasions are, the dye will cling to the tissue and the corneal layer, and that just demonstrates where exactly we need to, see.
Explain why you want to do it with the mice first. I didn't,
Yeah.
That's where you were gonna start.
Yeah, so that was just outlining a potential experiment in which this could be worked in mice. It's not harmful necessarily, so it's just basically to get a baseline for how this would work. Obviously, because this has never really been done in combination before. So the mice would be useful because we can apply the corneal dystrophies and the aging of how it progresses over time in mice, and then we can understand. But since it's not like, it doesn't pose any harm necessarily, the mice would be a good baseline for data, but it really doesn't it wouldn't be like, oh gosh, the mice are dying,
Thank you. I think it's a fascinating proposal. So I have I'm curious about, given the geometry of the eye, which is kind of the ball, do you think that your confocal approach would be able to actually visualize any part of the eye, or is it going to be only localized to certain areas?
Yeah, so that's why we're using the fluorescent staining to really get a better understanding of where exactly the problem is coming from. So when you use the fluorescent staining, it highlights the issue, and confocal microscopy in vivo specifically, which means in a living being and connected to like a person, it's able to focus on any part of the eye, but it does take a bit. You have to like center it on where you want it to be. So by using fluorescent staining, we're able to accurately pinpoint where we want to look deeper into the tissue of the eye and the corneal layer.
If you go back to your last slide, actually, you are nicely showing the rays, right? So it's going straight into the back. How do you image let's say if the disease is developing, let me know in the back, at the bottom of the eye, can we think about it? Would there be a way to get that part of the, almost like the dark side of the moon?
Yeah, so what this image doesn't really demonstrate and what confocal microscopy, or at least in vivo confocal microscopy, is the reflection of the lasers and determining the tissue that's damaged. And so a more accurate representation of this image would probably be the lasers bouncing back and there being a detector to determine what exactly we're looking at in the tissue. And so it's pretty applicable to like the eye, like all parts of the eye, but specifically because we're looking at corneal dystrophy, we wouldn't really be looking at the dark side of the eye, you know, further back, because corneal dystrophy is focused specifically on the cornea.
Any last questions? Okay, great. Thank you, guys.
Okay, group 9.
Hi, my name is Galvin.
Hi, my name's Isha.
I'm Eugene, and this is our presentation. So our question is, how can quantum dots be used to better understand the development of autism? Autism is a condition that occurs during the development of a child within the womb and can greatly hinder the child and their families for the rest of their lives. We wanted to find ways to use quantum dots to improve or eliminate this issue to benefit the lives of these families. Our approach to finding this problem was to research how quantum dots work and what they are, and eventually we found ways to medically apply these particles.
And so the type of microscopy we're using is fluorescent microscopy. Fluorescent microscopy is a specialized optical microscopy technique that is used to visualize specimens that fluoresce, meaning they emit light of one color when illuminated with light of a different color. In this technique, fluorescent dyes or fluorescent proteins are used to label specific structures or molecules within cells or tissues. When these labeled specimens are exposed to light of a particular wavelength, they absorb photons and then emit light at a longer wavelength, which is detected by the microscope. And so we're using quantum dots and so quantum dots are semiconductor nanoparticles that exhibit unique optical electronic properties is due to their size and composition. The optical properties of quantum dots can be tuned by changing their size. This tunability allows quantum dots to emit light at specific wavelengths depending on their size, which is useful in applications such as fluorescent labeling and displays. And so the lack of fragile X mental retardation protein, FMRP, causes one of the autism spectrum disorders named Fragile X syndrome, FXS.
So one of our inspirations was this article where it was discussed how quantum dots were used to distinguish and label mineral stem and frog and nadir cells in mice embryos. The QD was injected into the uterus of the mother, and after the baby mice had formed, the data was observed. The data established the safety of the process, stating that quantum dots did not cause any NSPC differentiation, which an NSPC are the neurons that construct the brain as well as the spine. The main takeaway that we had from this article was that quantum dots could pass through the uterus of the mother without harming or attaching any of the cells of the mother, which showed us that quantum dots could reach the embryo or fetus safely.
So the second article we used
focuses on developing a multiplexed immunoassay biosensor for detecting serum biomarkers. And so the beta HCG and AFP, which are indicators of Down syndrome. The synthesized quantum dots were characterized using various different techniques such as absorption and photoilluminescence spectra and transition electron microscopy. The quantum dots demonstrated stable photoluminescence properties in both water and chloroform, and it was able to detect low levels of beta HCG and AFP with high specificity and sensitivity. The biosensor was able to effectively detect the serum biomarkers, providing a potential tool for early diagnosis of Down syndrome through non-invasive blood tests.
So our solution, as we mentioned that when the gene FMR1 is inactivated by a nucleotide repeat expansion, the lack of MMRP will results in the fragile X syndrome. And what we are proposing is that we're trying to use quantum dots to label the FMRP and FXS mice brains and compare it with normal mice brains to provide information about the difference of distributions and quantities of FMRP in different brain regions. And we are also trying to obtain data such as which brain regions have the most reduced levels of FMRP after getting FXS. So the procedure of the experiment would be firstly, we prepared two groups of embryo mice, and one group is normal, while the another group have FXS. And so how we get the FXS mice brains is that we used a kind of technique called Crelux-P. We actually bind a stop codon to the FMR1 gene so to inactivate it. But actually we don't do it ourselves. We just buy it from some biomedical companies. And the second step is that we inject the quantum dots conjugated with anti-FMRP antibodies. This is the type of immunofluorescence technique, and we inject quantum dots into the embryo samples in order to label the FMRP with fluorescence. And under optic microscope, we just observe the brain samples from two groups of mice at the embryo stage, juvenile stage, and also adult stage, and we record the images and we compare the difference of the distributions of FMRP in regional brains between FXS brains and normal brains.
So the reason we chose QD over other methods such as GFP, which is also another genetic tracking method, was that QD can cross-link with proteins while GFP can only fuse to the gene frame. This means that QDs can be targeted to specific issues and are better at labeling and tracking cells. Also, as a protein, GFP is not able to be attached to FMRP, which means that we can't actually use it for our experiment.
So impacts of our study is that by understanding the distribution of FMRP in different brain regions will help us learn about the specific brain areas affected in FXS, and it is crucial for scientists to develop targeted therapeutic interventions and improve the specificity of the current medicines. And the remaining questions are some questions being raised up by our study is that how does the distribution in FMRP and FXS compare to distributions in other forms of autism?
Thank you.
Thank you.
Questions from the audience, please.
Thank you. Please correct me if I'm wrong here as well, but I think quantum dots are usually composed of different types of metals. Using some extra methods as well. Was there any thought given into the potential toxicology impacts if you're trying to look at an in vivo study using quantum dots?
Yes, we did look into that. But the toxicity we found was not that much that it would cause a great effect to the health or the growth of the cells. So we felt that it would be like the other benefits outweighed the negatives.
Thank you. For like the presentation, I just want to ask, because like autism is a spectrum, so does it apply this like to like all the spectrum or just like a specific spectrum of autism?
Yeah, many types of syndromes of ASDs are like mainly caused by like certain proteins and certain genes, and we can use and also we can combine we can use quantum dots to label those genes and proteins.
Thank you.
Recommending like, I'm wrong, but I understood that you were injecting mice, right?
Yeah.
You want that. So, what I missed maybe is, are you proposing to do, the optical microscopy in vivo? Or, after sacrificing the animals, can you just elaborate on this?
Yeah, I mean, we are trying to do it in vivo, but we are still researching on some. We have studied the type of technique called two photons microscope that it could use to observe like thick tissues such as brain tissues, but I'm not sure whether it is possible to use it to do a vivo observation.
It is, but it's certainly challenging, especially imaging through the brain. I mean, through the skull, right? You have to go through the skull. So you spoke a little bit about two-photon microscopy with different soft tissues. There's even another technique called three-photons microscopy to go through the skull, right? So yes, it can be done. Yes.
Thank you.
Anyone else question? All right, thank you guys.
All right, this is our last group.
All right, last presentation. So we were researching.
Hi, I'm Robert. From Archbishop Meade High School.
I'm Harry,
I'm Hazel.
and we're researching talosins, and we're trying to see if they can enhance the efficacy of antibiotics battling against bacteria, specifically Pseudomonas aeruginosa strains. So Pseudomonas aeruginosa it's a bacteria, and it's multi-drug resistant, means that it's resistant to many antibiotics, including ceftazidime. Anyways, talosins they're bacterions produced by bacteria, and they offer a promise as an antibiotic adjuvant. And so we're gonna see if the talosins they can enhance the efficacy of ceftazidime, and then we want to see if it'll increase the effectiveness against the bacteria. And if this is successful and the synergy is there, then this can lead to new novel therapeutic strategies in medicine. And it's also a possibility that the dosage of ceftazidime can be reduced to help combat the resistance development of the bacteria. And so we're going to be using fluorescence microscopy to visualize the talosins. and we're gonna fluorescently label them to see their activity and their growth and production from the bacteria. And the field that we're gonna be doing this in is antimicrobial resistance research and drug discovery and development. And ultimately, we're targeting a bacteria that is known as a superbug because of its antibiotic resistance properties.
Okay, so it's important to recognize, first of all, that ceftazidimine is not a complete remedy for Aragonisa. It is more like a first-line defense against it. So Aragonisa is the root problem for many diseases like sepsis and pneumonia. Ceftazidimine is gram-positive and negative, meaning that it can go through thick cells, thin cells, and a lot of diseases. So it's used as generic medication. Third generation ceftazidimine does have some immunity against beta-lactamases and efflux pumps. So efflux pumps are basically, every cell has them. It pumps out toxicity. And beta-lactamases, you can think of them as like they provide warning for the cells. But phages can eliminate this, and specifically tailsins can. In 2021 study, tailsins formed a biosynthetic gene cluster that diluted antibacterial resistant diseases like Aragonisa through membrane proton motive force. You can imagine the membrane proton force as being a very it's like brute force, but with protons instead. Excellent. The way we're gonna the type of microscopy used in this would be fluorescent microscopy, and it works like this. Basically, there's a light source, and that light source is projected through a filter, and that filter right there, the excited filter, you could call it, is it selects a certain wavelength for that light, and afterwards it deflects towards a beam splitter, which picks that up into an objective, and it's absorbed by the fluorophores, and it's shot back to the ocular, where we can observe it.
Our solution, what are we proposing? In our research, we aim to investigate the potential of tailsins in a means to enhance the properties of antibiotics. We specifically fixed on considering that of ceftazidimine as a kind of treatment against multi-drug resistant P. aeruginosa strains. Further, as our proposition, we conclude using the identification of ceftazidimine modifications and combinations that are capable of enhancing the efficacy of antibiotic medication. In closure detail, this information offers insight into the resistant mechanisms and potential targets in future drug developments of antibiotics, along with advancements in precision antibiotics and their components, such as tailsin development. Our research will also aim to collect data on pharmacokinetics and pharmacodynamics and resistant profiles.
All right, so how does it work? First, we have to isolate and then label the tailsins, and we can do this through centrifuge to separate the tailsins from the bacteria. And then there can be other purification steps like chromatography and spectrometry. And then after that, we're going to test activity individually without the ceftazidimine against the Aeruginosa and Petri dishes to determine the minimum inhibition concentration, which is the minimum amount of tailsins needed to combat the bacteria. After that, we will combine the tailsins with the ceftazidime at various ratios, which is known as checkerboard dilution, so we can visualize growth inhibition patterns. And this can be seen through fluorescence microscopy. And lastly, we have to conduct time kill assays so we can measure the bacterial death over time. And this allows for rapid evaluation of tailsins, and it can allow us to see if it's a good booster for antibiotics against the bacteria.
Why is it better? In recent years, multi-drug resistant Pseudomonas aeruginosa strains have become increasingly common and often pose substantial challenges within a clinical and scientific setting, as as current treatments are extremely limited. By tailoring ceftazidime as a considerably well-known antibiotic, which is used for effectively combating such resistant strains like MDR, it provides a viable solution as opposed to the treatments on the market. The research we conduct contributes to growth and scientific knowledge by investigating resistance mechanisms and being capable of developing antibiotic formulas, which can further inform antibiotic development, not only for ceftazidime, but also assisting other antibiotics that face common complications with resistant strains. Future directions. Any case where synergy is possible and efficacy of ceftazidime is enhanced, this could potentially lead to the next step of drug testing on specimen, which would hopefully eventually lead to the public use of antibiotic drugs. If our hypothesis is proved wrong, questions that support separate dosage of both substance could be researched. For instance, tailocins could be injected, which would weaken the bacteria for an infection of ceftazidime at a later time, and the direction and discussion from the experiment is numerous and can vary in study. Scientists can develop more targeted and effective strategies to combat MDR and P. aeruginosa infections and other diseases. These are our references. Thank you for listening, and we'll start the Q&A.
Okay, let's muster up all our energy for our last group to have some great questions here.
Can you comment on the mechanism of action for ceftazidime and how or whether you would expect the tailocin to actually have some synergistic effects? You're proposing that it's going to be an adjuvant, right? So I'm wondering if what the tailocins do and how ceftazidime actually works actually overlap in any way, or if you're just sort of throwing two things at it and two things are better than one.
So the tailocins, they can, it's possibly that they can weaken the bacteria. So then the antibiotic can go in and actually do its job by killing bacteria. But that's a hypothesis.
Do we know how ceftazidime, what it does to kill bacteria?
Well, we don't know specifically, but it's an antibiotic, right? So the antibodies attack.
Kind of question to what Adam just asked. What would be your endpoint assay you're using fluorescence microscopy? What is it that you're going to be measuring?
We're going to be measuring if it can improve, like if it can enhance the performance of ceftazidime against the P. aeruginosa. And that's what we want to see if it can improve. If it kills more bacteria with the time kill assays, like measuring bacteria death over time.
So measuring death.
Yeah.
In that case,
Sometimes this antibiotic doesn't work. Like there are conditions where it doesn't work, and we want to have like almost 100% efficacy.
Are there any studies right now that show the efficacy of tailocins versus the superbugs that are out there? Like, because they currently exist today, right? And so I'm curious to know if there's been any studies kind of showing whether or not they are effective at killing some of the superbugs or weakening them in any sort of way.
What do you mean by superbugs?
So the antibiotic resistant.
Well, yeah, there are studies out there. We just didn't include them in our presentation, so we're not educated on that. But we could look into it, and we could definitely find it for you.
Any last questions? Okay, let's take our last group. Okay, so we're just gonna do a little bit of closing remarks. I wanted to see if Adam or Ron, you have anything that you would like to say to the students? Put you on the spot here.
Okay, I'm sorry. So, so to the parents, thank you for sending your students here. It's always great, especially now being back on campus full time with everyone coming here. We've been doing this program, I think this is the 9th year. And as Shailene mentioned, we've been running summer programs for 16 or 17 years. And it's really always fun to have a new, young, vibrant group of folks come together and really take in the fire hose of information that we present them. I want to let all the students know that we asked you to do a non-trivial thing, right? Shailene was joking about, the challenges of coming up with an original research concept that no one's ever done before and do that in a matter of 3 days, basically. And I know it sounds easy, but this is something that I spend my life doing. This is what scientists do. It is non-trivial by any stretch. And I'm just impressed that you guys were able to take in and integrate as much information as you have in such a short period of time. You've got a really great group, lots of great questions, good energy. I know I had to sort of draw it out of you the first couple of days every morning with the good mornings, but it's really fantastic to see you all come here, and we're really excited for whatever comes next. You all know where to find us. Maybe some of you will end up at UCLA. That certainly happens. We have lots of times where students from these programs end up here and stumble by and bump into somebody that you met 5 years ago. So hopefully we'll see some of you soon. And hopefully you learn something interesting that will, catalyze some, new interests wherever you lead wherever you head next. So I'd just like to say thank you all for coming. It was a great week. And answer on behalf of everybody that I think this was a really, really great program this year. And looking forward to, of course, what's next for you. And I'll say any feedback that we can get to improve the course would certainly be welcome. So thanks so much. And enjoy the rest of your time.
I have something to say. Quick introduction for people that don't know me. Hi, my name is Ribose. I'm the Director of Education here at the CNSI. It was really fun getting to be with all of these students this week. We've heard a lot of thanks for this program, but there's one person that has not yet been thanked, and that's the person that is to my left, which is Dr. Shailene Tanimoto, our education manager. And from start to finish, a lot of this was very much her leadership and her production and getting all the moving parts that needed to come together. So Shailene, thank you so much for steering this ship and being the shepherd of all of the various sheep that go along with doing this program. Thank you.
Okay, so I just again want to reiterate what Adam said in thanking our parents, and I'd like all of the students right now to look or think of your parents, because this is not a cheap program. I don't know if y'all are aware of that, but this is not a cheap program, so there's that aspect. But then they're also fostering your growth and encouraging you guys to pursue these things. And so I think many of us who run this program wish we had something like this when we were in high school, so that we could have started earlier in our science career. And so this is a great start for you guys, whether you're interested in pursuing this topic or not. At least you were exposed to it and you learn a little bit more about yourself and what you're interested in. So again, let's thank our parents, please. Thank you. We love and appreciate you, all the parents out there. And then, like Adam said, we take feedback. So students, if you could fill out this survey, on SurveyMonkey, it's just like quickly rating 1 through 5, how you felt about the pace and the content and all of that. So we would love your feedback so we can keep improving this course every year. Okay, and the last thing is that we're having a reception up on the 6th floor patio. Everybody's invited. Students will get t-shirts, free t-shirts at the end, CNSI shirts. So thank you everybody. Thank you for coming and thank you for another great year.